All right, today we're going way, way down to the nanoscale. We're talking about DNA origami and cracking a code that could totally change how we build, well, just about everything. Think tiny robots, powerful new computers, you name it.
So, you know how scientists can take these long strands of DNA and fold them into super precise microscopic shapes? It's called DNA origami, and it's amazing. The potential is just huge. But yeah, there's a big problem, a really big one that's been holding the whole field back.
And here's the problem in a nutshell. What you design on the computer isn't always what you end up with in real life. I mean, look at this. On the left, you've got this perfect, beautiful digital blueprint. But on the right, that's the messy reality of what actually comes out of the test tube. So, what gives? Why the huge difference?
And this isn't a small challenge. It's staggering. The researchers put it this way. For any single shape you want to make, there are pretty much infinite ways to write the folding instructions. It's like having a million different recipes for the same cake. It's a total paradox of choice for the designers.
So that really is the central mystery here. How can two designs that look almost identical on paper give you these completely different outcomes? One comes out perfect, and the other is just a useless clump of DNA. Let's figure this out. Okay, so to crack this code, some brilliant scientists at the National Institute of Standards and Technology came up with a really clever experiment. They decided to give the DNA a kind of nanoscale stress test.
They didn't just test one or two designs. No. They made 34 different versions of the exact same rectangular shape. And each one had just a tiny little change in its blueprint. You know, like how the edges were stitched or what was holding it together. They basically wanted to push them all to the breaking point and see which designs held up and which ones just completely fell apart.
And this is where it gets really smart. To see what was really going on, they didn't use pristine, perfect lab conditions. Instead, they used what they call an accelerated stress test. They basically made the folding process harder on purpose. Why? Because it magnified every little flaw in the designs, making it way easier to separate the winners from the losers.
And wow, the results were dramatic. Just look. On the left, you've got design number 1, total success story, clean, perfect rectangles. But on the right, design number 7. The blueprint was almost the same, but the result was a complete, catastrophic failure. So, what in the world is going on? What are the secret forces at play here? Well, it turns out the answer has to do with something called cooperativity. The researchers figured out that there are basically 4 secret forces of cooperativity that control this whole assembly process.
So what is cooperativity? Think of it like teamwork, but on a molecular scale. When one tiny part of the DNA structure folds into place, it can either help its neighbors fold correctly, or it can get in their way. It's this whole complex dance of pushes and pulls happening all at once.
And these forces, they break down into 4 main types. You can see it in these diagrams. Some of these interactions are good. They help push the assembly along. But others, they're bad news. They create roadblocks and cause things to mess up.
Okay, let's quickly run through them. First, you've got the good guys, things like a helping hand or teamwork. That's where one correct fold encourages its neighbors to do the same. But then you've got the villains, like the roadblock, where some other random DNA strand can just float in and stop the whole process cold. And finally, you've got something called sticky ends, which is like a little energy bonus that helps lock everything together.
So, okay, now that they'd identified all these forces, the researchers got to the really big question. Could they actually use this knowledge to predict which designs would work and which would fail?
Naturally, the first thing they tried was to see if any one of these forces was the magic bullet. You know, was it all about maximizing teamwork or just about minimizing roadblocks? Could it be that simple?
Yeah, no, not even close. When you look at any single force by itself, the data is just a mess. A total chaotic scatterplot. There's absolutely no pattern, no trend. It was pretty clear the answer was going to be more complicated than that. But then, this is where the real breakthrough comes in. They thought, okay, what if it's not about any one factor? What if success is all about the right combination of them?
And there it is. Look at that. They built a model that combined 3 key things: thermal stability, that helpful teamwork force, and the bad roadblock force. And just like that, all the chaos vanishes. All of a sudden, you see this clean, predictive line. The secret wasn't one single force at all. It was the balance between them.
The best way to think about this model is like a weather forecast, but for DNA. For the very first time, scientists can punch in a design and get a really reliable prediction,
Is this thing going to assemble right, or is it going to fail? This could save just an unbelievable amount of time and money spent on trial and error.
So what's the big takeaway here? What does this mean for the future? Well, this breakthrough hands us a brand new, powerful set of rules for actually designing DNA nanostructures that work.
And the rules are actually pretty straightforward. Keep your staple strands short. Make sure your seams are symmetric. Keep any extra DNA junk on the outside of the main structure. And what's really cool is that our very human aesthetic preference for things that are clean and symmetrical, turns out that's actually great advice for good design at the nanoscale. Who knew?
The researchers themselves have this great metaphor for it. They say it's like they've cut the Gordian knot of DNA design. For years, the field was stuck on these three huge problems. Which forces actually matter? How do you even measure them? And then how do you combine all that data into something useful? This new model basically solves all three of those problems in one go.
And the practical impact? It's huge. Remember that harsh stress test they used? Well, if you apply these new design rules under normal, commercial-style conditions, you could make the assembly process over 1,000 times faster and more efficient. That's a game changer. But honestly, this is just the beginning. The researchers point out that the real power of this model comes next. Because now, they can look at the few designs where the model still gets it wrong and use those failures to figure out even deeper, more fundamental rules of physics. In science, every failure is really just a clue pointing to the next big discovery.
And that leaves us with one last, really exciting question. For so long, the incredible promise of DNA technology has been kind of stuck in this trial and error phase. But now that we actually have a blueprint to predict success, what are we finally going to build with it? What incredible nanoscale machines are now possible?